Hello everyone, welcome to a past paper review. Today I'm reviewing a CAPE Biology paper 1 and this was from the year 2019. Now before we start this examination, please remember to read through the instructions carefully and also ask any questions if you're in doubt before the examination begins. Now let's jump into the first question, and this question reads, which of the following molecules best represents the structure of a triglyceride? And once you talk about triglyceride, what you need to think about are lipids. Lipids which contain 3 fatty acids and a glycerol. That's why the word triglyceride comes in. Now let's examine each of the structures first before we look at the exact answer here. And so in option A, this structure is a nucleotide, and I want to specify why this is a nucleotide, so just hold on a little bit. The nucleotide. All right, and so it's a nucleotide because it contains a phosphate group, which I'm circling right now. It also contains a 5-carbon sugar, which is a pentose, and there are 2 types of them that are found in nucleic acids or nucleotides. And they are either ribose sugar or deoxyribose sugar. Now the ribose sugar, it is found in the RNA, and the deoxyribose sugar is found in DNA. Now to be specific, this part here is a nitrogen base, and this nitrogen base is actually for adenine. So let's put that name right there. So this is for adenine. All right, and adenine is found in both RNA and DNA. For structure B, this is a polysaccharide, and this is a polysaccharide. One thing that make you sure about polysaccharide, because you see many OHs as well, and not just the OH, but also you see the hexagonal shape, and the hexagonal shape which represents 6 carbon. So I'm going to show you the 6 carbon just a minute. So this is a polysaccharide. All right, and so the 6 carbon, you have 5 carbons actually in the hexagonal shape because an oxygen is there. So there are 5. 1, 2, 3, 4, 5. And the 6th carbon is right here. All right, so definitely here you notice the many glucose molecules and other factors are pointed out. All right, so that definitely a polysaccharide. Now for diagram C, this is actually answer, so let's circle that as the answer. And why this is the answer, because right here represents a glycerol, and attached to the glycerol you have 3 fatty acids. So there's one right here, there's another fatty acid right there, there's another fatty acid right here. How we know is a fatty acid? Remember the carboxyl group COOH, all right, but the R group will be attached to that, which is a side carbon chain. All right, so you have a side carbon chains attached to that to be as long as based on the type of fatty acid, all right, whether it be saturated, unsaturated, and so on. All right, so now let's go into structure D. Structure D is a phospholipid. So let's write that name in. Is a phospholipid. All right, and I'm going to tell you why it's a phospholipid right here as well. It's a phospholipid. All right, and it's a phospholipid because it's similar to structure C, which is a triglyceride, which is just a lipid. And so phospholipid because one of the fatty acid is replaced by a phosphate group. So that's a phosphate group. And you have 2 fatty acids there attached to the 1 glycerol. So you have a glycerol, 2 fatty acids, and a phosphate group. So that is a phospholipid. And you need to also remember that phospholipids are found in cell membrane or plasma membrane. All right, let's go to question number 2. And this reads that item 2 refers to the following diagram of a protein structure. Notice there see an amino end right here and a carboxyl end right here. All right, and you notice the R groups are there, which is the side carbon chains. All right, so now we can also even think about the number of amino acids present, because the R groups, they're numbered. So this will be 1, 2, 3, 4, 5, 6 amino acids actually make up this protein structure. However, let's go to the question. It said, during hydrolysis of protein structure shown above, which of the following bonding interactions is broken? Now, the first thing you need to think about what the word hydrolysis means, and hydrolysis actually means adding water to break, because lysis means to split or to break, hydro mean adding water. So, along with this concept, what you also need to remember is that the amino acids are joined together by the called peptide bonds. And so to break the protein structure into various amino acids, you must break the peptide bonds. And so the bonds that will be broken here will be what? The peptide bonds. All right, so question number 2, the answer is B. For question number 3, it states that a diet high in triglycerides, which is a fat again, which is lipids, right, may lead to obesity. Now, it said the feature of triglycerides, which most likely contributes to this, is that they, and option A say, are insoluble in water. To be insoluble in water is really a good choice, but we want to see if there's a better choice there. But let me point out real quick, to be insoluble in water, it therefore means that the structure cannot be easily broken down by hydrolysis. In fact, it's not broken down by hydrolysis, because if it's insoluble in water, it can't react with water to be broken down. So what you need to remember here now, that this being insoluble in water will allow the structure to store a lot of energy. So it's mainly for storage. So being insoluble, the structure itself is a good storage structure within the body. Now, having many CH bonds, which is very good, because the more CH bond is within the triglyceride or in the fatty acid, is the more complex that fatty acid is. And being more complex, it means therefore it will even store in a very large quantities, and therefore could lead to obesity. And so the best choice right here between A and B is actually B. All right. Content of triglyceride is not the reason to lead to obesity, definitely, because of course, you know, the glycerol part is not the main concern in terms of being unhealthy. It is the fatty acid part. And remember, the fatty acids could be either saturated or unsaturated. And to remember what defines them in terms of saturation. The double bonds will make them unsaturated, but the single bonds, only single bonds, will make them saturated. And so having many OH group is not correct, all right, because actually OH group will make them be reactive, in fact, and allow them to break down. So that cannot be the answer there. Now, question number 4. In question number 4, it says, which of the following structure is not likely to be seen in an electron micrograph of a plant cell? And so I want to point out quickly, based on these options, we have here, we have centriole. And centrioles are not typical of plant cells. That's the first thing you need to remember. Centrioles are not typical of plant cells, but they are seen during cell division of animal cells. So looking at the plant cell, you not typically see centrioles. So centriole could be a good answer right there. Let's look at tonoplast. Now, tonoplast is a membrane that surrounds the large central vacuole in the plant. So definitely you will see that, because it's, the vacuole will be obvious. And you will see the tonoplast definitely. Ribosomes, which will be the organelles responsible in terms of making protein, you will see also those. Golgi apparatus, which is packaging and transporting of substances, you will also see those. So the best answer here, which is not likely to be seen in the micrograph, it is the centriole. All right, so great. All right, let's go to question number 5. In question number 5, it said, what is the highest level of protein structure exhibited by hemoglobin? And so the first thing you want to remember now is the order of which proteins are folded. And the folding of proteins start from the primary, secondary, tertiary, and quaternary structure. So these are in correct order. But I want to point out something here before I even go further. What is actually represented by these structures? And so when you talk about the primary structure of protein, what you're talking about is the order of the amino acid. What do you mean by the order of amino acid in terms of which amino acid will come first, second, third, and so on. Okay, so this represents the order of amino acids. All right, and so when we talk about the secondary structure, what you need to think about here, talk about it is the helix form of the protein folding. In this point is when you have the twisted arrangement because of the attraction or the bonding between hydrogen. So because of hydrogen bonds. So here we're going to have here helix structure or the helix form of the protein. When we talk about the tertiary level, you're talking about the 3D model or the 3D shape or orientation of the protein itself. So this would be a 3D orientation or shape of the protein. The quaternary structure, this is where you'll see here, you see many polypeptides. And so this is where you have the more complex or the most complex structure. So here will be many polypeptides. All right, so many polypeptide bonds will be there. And so option D will be our correct answer. As I said, this is the most complex structure in terms of a folding of proteins, and hemoglobin is a protein. Now, question number 6. It said which structure is found in both a prokaryotic cell and an eukaryotic cells. So prokaryotic cells are very simple cells, eukaryotic cells are more complex cells, and prokaryotic cells are typically found in bacteria. Now, eukaryotic cells are generally or typically of multicellular organisms. So let's talk about the structures now that you'll generally found within both of them. And so when you think about ribosomes will make protein. All right, and so you may find that in typically all cells. And I'm going to give you some other things. So let's hold on to A bit. Chloroplasts is not, because chloroplasts are found within plant cells or plant-like cells or plant-like organisms. And mitochondria will produce energy, and not all cells contain mitochondria as well. Nuclear membrane, prokaryotic cells do not contain nuclear membrane, definitely. So ribosomes will be our correct answer. And I just want to point out something here as well, that cell membrane or plasma membrane must be in both cells. In fact, all cells must contain a plasma membrane, right? And also cytoplasm and DNA. So the common structures for all cells is ribosome, cytoplasm, cell membrane, and DNA, or at least chromosome. All right, question number 7. It said collagen exhibits a secondary level of protein structure and organization because, I know we're talking about here now, remember this word come back, secondary structure. What secondary structure is, again, remember we say this is where the helix form, right? And so what you need to think about is helix. Secondary deals with helix, as I mentioned earlier. It contains 4 polypeptide chains. And once it's a polypeptide chain, you're talking about here, it is a quaternary structure. So this could not be the answer. A could not be the option, okay? Now, each chain forms a helix. And now this is good because the word helix is there, but it's held by covalent bonds. Let's examine it a little bit. Now for C, it said, each chain forms a helix, which is also great, but held by what they call hydrogen bonds. And I also talked about this earlier because the bonds, matter of fact, the hydrogen bonds is what will cause the chain to form that helix structure. So C will be a better option than B. Okay, so C is good. All right, and let's look at D. It's made up of repeated units of 3 different amino acids, and this is not true. All right, so this is referring to the primary structure to talk about the order in which the amino acids will fall, refers to the primary structure. So definitely option C is our correct answer here. Now for question number 8, let's go to, yep. All right, here it starts. It's item 8 refers to the following diagram, which shows a transverse section of a plant structure. And so before I go into anything, let me quickly label these structures for you real quick. So here, the end, these small things that protruding, those are the root here. Okay, that's the root here. All right, and then we have All right, you know what? Let me just quickly draw some lines so it'll be easy, I think. All right, so we have X. Let me just put this in. X is the phloem. All right. And the structure in the middle represents the xylem. Let me put that on, then I will label it. So that will be the xylem vessel. And then we have here, which is a cortex. Let me just write it right there. So we also have the cortex. I'm going to put the lines there so it'll be easy for you to see exactly what I'm talking about. That's the cortex. And then we have this one in the middle right here. This represents is our endodermis. That's our endodermis. Okay, so let's put the labelings there. So endodermis is here. Our xylem is this structure right here. And our cortex is right here. And of course, these are the protruding lines. They are root hair cells. So those are root hair. And they contain root hair cells. All right, and generally typically for absorption. So the phloem is X. So it said the area labeled X represents what? And so X has to be the phloem. Okay, that's not what it is asking here. It said it represents, it didn't give a name. So let's re-examine this question. It said the area label X represents a cell, which is not true because the phloem is not a cell. It's a tissue, which is great because the phloem is a tissue. Okay, yes, so this is our answer right here. Not an organ, not a system, but definitely a tissue. But it is a phloem, xylem, and you have the phloem tissue and the xylem tissue. Okay, excellent. Now, question number 9. And for question number 9, it reads now that Bobby receives a tropical freshwater fish for his birthday. Before I even go any further, look at the statement. It says freshwater fish. So this is some indication of something special about this fish, a freshwater fish. Just to highlight that a bit, okay? It said while experimenting with the fish, he places it in seawater, poor fish, and it dies. Oh my God. Anyhow, it said which of the following statements most likely explain the reason for its death? So first, let's think about the scenario first. If the fish is in fresh water, it therefore means the fresh water is isotonic to the fish, right? Because it is natural environment. Putting the fish in salt water now, because they put it in salt water, by placing seawater, which is salty, right? It's seawater, it is salty. So therefore now means that the seawater is hypertonic to the fish. So once cells are placed in hypertonic solution, let me just make sure I write that once you see which one I'm talking about. Not hypotonic, but hyper. The fresh water is isotonic to the fish because it is natural environment. So let me just quickly put that in. Okay. So the fresh water is isotonic. Let's put it on this side right here. No, let's put it right. I could put it right here. So the fresh water is isotonic. I want you to see that. Mean that the concentration is the same. Okay. Natural environment, isotonic. But the seawater now going to be hypertonic. Okay. Let's make a note of that. All right. And so once cells are placed in hypertonic solution, the cells will lose water and become flaccid. All right. And so let's look at what will happen here. So water diffuse into the fish, which is not true. Because if it was hypotonic, then this will be true. So A is incorrect. It's a water diffuses out of the fish, which is true. Water diffuses out of the fish because it's hypertonic. Let's see if there's any other options that even better than this. It's ions diffuse from the fish to the seawater. Not true, because the ions will not move. Water moves through osmosis. Less oxygen in seawater than freshwater. No, absolutely not. All right. So option B will be the best answer right here. Water will leave from the fish. So the fish will lose a lot of water, and the cells will be flaccid. All right. So now let's go to question number 10. For question number 10, it said the following table shows 3 processes involved in the movement of substances across cell membranes. It said which of the following rows correctly classifies each process and as active and or passive in nature. So which is active, which is passive. Before we go any further, let's quickly just make a point right here. Once talking about active transport, you're talking about the transport that uses energy or requires energy. So let's say needs energy, okay? The needs energy. All right. And if you're talking about passive transport, you talk about there's no need of energy. All right. But there's some other stuff I want to talk about. So passive means no energy. All right. So why energy is needed? Let's talk about that as well. And so for active transport, what is also happening is that the solution or the solvent from the solution will move against a concentration gradient. So the movement will be against the concentration gradient. So against the concentration, I'm just put CON as short for concentration gradient. And also what is important as well is that the solvent will move from low to high concentration, which is not the usual way for movement of substances. So move from low to high. So since move from low to high concentration, it therefore means energy must be required. If you move into somewhere that is already concentrated, you have to put some force into it. Think about you going to a room that is already packed with people. For you to enter that room, you have to force yourself in. So the same thing with active transport, moving from low to high, you have to force that molecules against a gradient, and so therefore to require some energy. But talk about passive now, the opposite is true. So you go along a concentrated gradient and also move from high to low concentration. Let's put that real quick, right? So here we have along a concentration gradient. So along a concentration gradient, and not just that alone, it also moves from high to low concentration, right? High to low concentration. All right, so that's a typical way which we will explain passive and active transport. Let's put it right here for your easy viewing. All right, great. So let's talk about which of these are generally passive, which are generally active. So osmosis and diffusion, if you think about the definition for the words, particles move from high to low concentration. So they are passive. The difference with osmosis and diffusion is that osmosis must go across a membrane and it must be the movement of water. But diffusion now could be movement of any particles from high to low concentration, right? And so both of them are passive. It's only that different substances are moving. But when it comes on to endocytosis, endocytosis is active. And so then our answer here will be C. So let's say option C is our correct answer for question number 10. All right, let's go to question number 11. And for question number 11, it said that 4 cylinders of equal mass are cut from a single potato. So you have a potato cut into 4 equal pieces. 1 cylinder is placed in distilled water. That means there's nothing in that water at all but pure water. And the others in 0.2 mole, 0.4 mole, and 0.8 mole sucrose solution. So in other words, we're talking about sugar. So they're placing some molar concentration of sugar. Now, what I want to point out right here is that the higher the concentration in terms of molarity, it means the higher amount of sugar present within that solution. Okay, so that's very important to note. It's that each cylinder is weighed at half-hour intervals for 6 hours. The graph below shows the results obtained. Which curve most likely represents the 0.4 molar sugar sucrose solution? So which of them will have the highest amount of sugar, or display the graph that the potato is placed in high sugar content? Now, what I want you to think about, the higher the sugar content is, the more the potato will lose water. Okay, that's the first thing you want to think about. Okay, so if you're losing water, it therefore means that the mass will decrease. The lower the sugar solution or the sugar concentration, mean less sugar in the water, mean that it gain more mass. So distilled water will move into the potato cell because it will be a hypotonic solution and cause the cell to become turgid. More sugar added, it therefore means the cell will lose water and the cell will become flaccid or plasmolyzed depending on the concentration of the sugar. Now let's think about this now. For 1 and 2, they're increasing. They're increasing in terms of mass. A change in mass, they're positive change. that means there's an increase in the mass. So these cannot be the correct answer, right? Because the 4 molar, not 4, 0.4 molar is the highest one. So remember, let's go back again, just so for you to make sure you see this. We're moving from distil, 0.2, 0.4, and 0.8. Looking for the 0.4. So let's see what may happen here. So one could be distilled water, right? So let's kind of put them there. So the one could represent distilled water because it is very high in terms of the greatest increase. So this could be distilled water. All right, so that could be distilled water. And 2, again, will be the next level. So the next level here will be 0.2. So this represents the 0.2 here. All right, and then you go down now to the 0.4. So this will be 0.4, start to decrease. All right, and then 0.8 will be the one that caused the most decrease, so 0.8. All right, and so we can assume now that graph 3 or line 3 represents our 0.4. All right, with this question, there's something I want to point out here. So our answer here will be C. Yep. Let me point out something real quick. Let's say this question asks you what molarity or sugar concentration could be isotonic solution, which means it'll fall somewhere right here, right? So if it fall here, there's no change for isotonic solution. What could fall on that line to cause no change between 0.2 and 0.4 could be 0.3. So 0.3 could be an isotonic solution. Let me put that in red just to make a note of that, right? So that's not given in the question, but I'm saying if they could ask you for isotonic based on this same graph, and isotonic will be about 0.3 molarity, if you should assume in terms of intervals, all right? All right, great. All right, let's go to question number 11. No, that's actually question number 11. Question number 12. Let's go to question number 12. And the question number 12, it said, which of the following correctly describes the importance of cholesterol in a cell membrane? So first thing, when we talk about cholesterol in cell membrane, what you need to think about is that cholesterol helps the cell membrane to become thick, and also in terms of the fluidity of the cell, okay? And so any answer that talk about thickness will be, or allow the cell membrane to be thick, and also help to maintain stability, or also help to maintain the cell itself. That's what the purpose of the cholesterol in the cell. So it's interact with fatty acid tail to exclude water, but it's not true. All right, because fatty acid definitely no, it will not work like that, because fatty acid is hydrophobic. Just to make a point right there. All right, it said it forms channel in the membrane, facilitating the entry and exit of polar molecules. Not true at all. All right, and so it has branching carbohydrate chain, which acts as identity markers, which is not the correct answer right here. But this one says it enhances the flexibility and mechanical stability of the membrane. Yes, because again, it helps with the thickness and the fluidity of the cell membrane. All right, so question number, what question is this? Question number 13. All right, just let me clear this 0.4 right here and see if I could take it out. Okay, 0.4. This is question number 13. I'll put 0.4 right here. Okay, that was our answer there. All right, so for this here, now it says, which of the following is the best example of anabolism? And so when we talk about anabolism, just to make a note right here, anabolism means to build up substance in forming more complex structures or substances. Catabolism means to break down. So catabolism, let me just make them right here. So anabolism means to build up. Let me just put it right here for you. Okay, so anabolism means to build up. And so when you see the word catabolism, now let's put it at the bottom right here. So catabolism means to break down. All right. All right, so that's very important for you to note right there. So catabolism refers to break down. Anabolism means to build up. So think about the word C, cut. So cut up in pieces. So if we break up polymers into monomers, that's catabolic reaction. To build up monomers into polymers, that is actually the anabolic reactions. So he said, but the word here say best now. So let's see what's happening. Glycogen convert to glucose, that is actually catabolic reaction right there, right? Because glycogen is a stored complex carbohydrate in the liver. All right, it's a peptide bonds are hydrolyzed by peptidases. So therefore means that the protein structure are being broken down by the enzyme peptidases. And so therefore this is also catabolic reaction as well, because it's broken down. And you see hydrolyzed, which means broken down by adding water as well. Amino acids are joined by peptide bonds to form polypeptides. And I like this here because amino acids are joined by peptide bonds, and amino acids are definitely joined by peptide bonds. And then it eventually forms into polypeptides, will form a protein structure. But let's see if there's any other options here, because the word here is best. Now for part D, now it said amylose and amylopectin combine to form starch molecules, which is also anabolic. So we have between C and D. But in terms of best, in terms of anabolism or anabolic reaction, the amylose and amylopectin combine to form starch granules is best. Why it is the best one? Because here we can see two different substrates being used, which is the amylose and amylopectin. But in the amino acids now, because it is not specified in terms of bonding, and so here we have peptide into polypeptide, is therefore means that the bonds were already formed, right? So all is happening into the structure is that the structure is getting more complex, not necessarily building on more amino acids, right? So if it's say amino acids in forming proteins or just forming peptide bonds, then that may be the best option right there. All right, but in terms of this, two different substrates coming together to form a more complex structure, definitely we go with that one. So D will be your preferred answer. All right, let's go to question number 14. And we have 45 questions in this paper, so we have a way to go. All right, I'm going to see how quickly I could move on. All right, so question number 14. It reads that item 14 refers to the following activity curve, which shows the effect of pH on the activity of enzymes. And so remember that enzymes are proteins, just to point that out real quick. And it said this is the rate of the reaction going up on the y-axis, and we have the different structures here, our different graphs, 1, 2, 3, and 4, and we have the pH level. It's a pepsin. So for question number 14, state that pepsin is an enzyme found in the human alimentary canal. And we know where this is found. It's found in the stomach. It's a which of the activity curves above best represents the action of pepsin. First, you need to note is that pepsin works in the stomach. In the stomach contains hydrochloric acid. Hydrochloric acid is a strong acid, and a strong acid will range anywhere between 0 to about 3. Now, above 3 going to 4, 5, and 6, those are weaker acids. So, looking at the graph carefully, you notice that here will be your strongest acid being demonstrated. 2 will be weak acid, all right? And so 3 and 4 are out of the picture. No, matter of fact, not necessarily 3. Let me just erase that, because 3, let's point out something right here. Look at the greatest activity for 3. It is less than 7, a little bit less than 7. So if less than 7, it's also a very, very weak acid, right? And so definitely option A will be a correct answer, because graph 1 represents a strong acid, a pH anywhere between 0 to about 4, or say 3.5 really, is strong acids. Above that tend to be weaker acids. 4 is actually alkaline or base, right? Question number 15. Let's go to question number 15. Let's see if I could speed up the time here now. In question number 15, it said item 15. Let me go back a little bit more. It said item 15 refers to the following graph, which shows the progress of an enzyme catalyzed reaction with increased temperature. And as you know, again, this is an enzyme. As temperature increases, enzymes tend to denature. All right, so here, if you notice at X, the line drops. So let's talk about what is happening here. It said the decline in the rate of X is most likely because of what? We know because the enzyme is denatured, but what will cause the enzyme to denature? That's the first thing we need to figure out right here, right? All right, it's the peptide bonds are broken. That is not denaturing necessarily. Well, the structure is actually changing if the peptide bonds are broken. But there's something specific we need to figure out. Let's just hold on to a bit. It's the enzyme loses its secondary structure. Not necessarily quite, because the secondary structure is just where you have the helix. Is not determining the properties of the protein yet, or the protein structure of the enzyme yet. It said the enzyme loses tertiary structure, which is a very good answer right here. But let's kind of think about it first. In terms of the tertiary structure, the tertiary structure determines the function of the protein. And when we talk about the function of proteins, you're talking about the shape of the proteins. And when we talk about shapes of protein, you can talk about the active sites of proteins. And remember that enzymes work or carry their reactions at their active sites. So this, look at the coolest answer right here, C. I like A because the peptide bonds are broken, which means the structure is actually changed. But being that the peptide bonds broken, not necessarily indicating the property of the protein structure itself necessarily. But C is more specific in terms of forming active site. If that active site changes, then the substrate can't react with that enzyme at all. So C is the correct answer. All right, so great. Temperature has nothing to do with the concentration of the substrate itself. Whatever substrate you put in, that's what will be there, unless it's a gas that will evaporate. And of course, you know, it will not be in gaseous form, right? Right, so cool. Number 16. Let's go to 16. 16 said, which of the following is found in both DNA and RNA? This is a very good question right here, yeah? Uracil is found only in RNA, right? Remember that. Uracil is only in RNA. Let's kind of mark that down. The uracil is RNA. You know what? Let me type this in. I may not be the best with this manipulation of writing tool, but just type it in real quick. Uracil is RNA, okay? RNA only. Ribose sugar, again, as I mentioned this before, is also in RNA. Okay, deoxyribose is in DNA. Just to make a note of that. Sugar phosphate backbone. Yes, this seems good. Yep, both of them do have a sugar phosphate backbone, but the sugar phosphate backbone is different in each, but they do have a sugar phosphate backbone. If it's an RNA, then the sugar phosphate backbone will be a ribose. And if it's a DNA, then the sugar phosphate backbone will be a deoxyribose sugar. Both of them are double helix. No, double helix is only found in DNA, so this is only specific to DNA. All right, so both of them do have helix, right? But double helix is only for DNA. Single helix is for RNA. Okay, so definitely our answer here is going to be C. Yep. This is cool. All right, question number 17. It said, question number 17 refers to the following diagrams of molecules which are involved in the formation of nucleic acid. All right, so nucleic acid here is a keyword. Nucleic acids. So nucleic acids refer to DNA and RNA, right? That's the first thing to remember. Now, the idea of these molecules, let's just talk about them right here. Let's go back to the question. So let me just read something again, just to make sure. All right, it said, okay, there's no question there. The question, I think, is the bottom of this, I think. Yeah, let's go to the bottom and see what they're asking here. All right, it said, which two molecules, when combined with a phosphate group, forms an RNA nucleotide? Looking for RNA nucleotide. All right, so let's look at the structures now. First thing that I'm going to take out here to eliminate this thing is the thiamine. Thiamine is only in DNA. All right, let me put that in. Thiamine is only in DNA. In matter of fact, I could say thiamine replaces uracil. Okay, where uracil is, that's where you find thiamine. So this is DNA alone, yeah? Uracil is in RNA. So you know that uracil will be one of the structures, right? So 3 will have to be one of the structures. 4 cannot be one of the option. Cannot, because the DNA, thiamine, DNA. Now let's go to the top here now. Oh yes. These now are our 5 carbon sugars. 2 of them together. Now, what is so important with these 2 structures? What is the difference though? Do you see any difference? There is a difference. Notice you have two OHs here, right? You have an OH right here and an H right here. So which one is which? Think about it. This one is quite oxidized. So we could call this now our ribose sugar. I just like the word to use, oxidized, right there to make it easier for us to understand. Now, notice an oxygen is missing. If the oxygen is missing at this H, it therefore means it was deoxidized. So therefore, this sugar here is a deoxyribose sugar. I like this. This one is deoxyribose. Deoxyribose. Yeah, this is great. This is a good paper. So now we know something here, that deoxyribose sugar is found in what? DNA. While the ribose is found in what? RNA. So here we have our indication already that, hey, these two structures, which is the ribose and the uracil, must be in RNA. Right? Must be in RNA. So therefore now the deoxyribose is DNA, and notice the name again. Thymine is in DNA, so deoxyribose. All right, so yes, our answer must be 1 and 3. Let's see what option is that. Oh, A is the correct answer. 1 and 3. All right, we'll speed it up a little bit now. All right, I just want to explain some of the stuff to you, so make sure you understand. All right, so number 18, item 18 refers to the following table, okay, which shows the codes and the matching amino acids. And so this is a part of the codon chart. All right, and so this is only a part of it. You could have a full chart, and you know, don't try to remember the whole chart, to be honest with you. There is no need for that. All right, so whichever following amino acid sequences would be produced from information coded on the DNA strand shown below. Now, they could ask, what strand is this though? You know, they could ask if this is a DNA or not. How know this is a DNA strand? T is present, thymine is there, right? So if thymine is there, you know that's a DNA strand. Anyhow, let's work this out real quick. Easy way to do this, or the easiest way to me, a matter of fact, what I will do, I'll do a conversion with this DNA strand, because when you're gonna transcribe it, you're gonna transcribe the strand, right? And then you translate this strand. So in terms of transcription, what I will do here now is to form the code by changing it into what they call the mRNA. So I'm gonna change this into mRNA. All right, so mRNA, lowercase m, RNA, which is a messenger RNA. What you're gonna do right here is to rewrite these codes, and remember they occur in groups of 3s, okay? So each 3-letter represents a codon. So what I'm going to do is to take the first 3 letters and rewrite them. But what I'm going to do, since I'm writing RNA, T is not present in RNA. So wherever you see T, going to change to U, because uracil will replace the thymine in the DNA structure. So the first thing is transcription. You get an mRNA strand. So first I'm going to say A. I'm going to change my T into U and say G. So follow me carefully. A-U-G. So the only thing I'm changing is the T's into U's. Please, I'm only changing T's into U's. So I'm having A-U-G first, and I go to the next 3 letters right here. I could mark them out as I go. So I'm going to, let's just split up into 3 easier, like this. So you're going to make it much easier on us. All right, so that's the first 3 right there. That's another 3 right there. Another 3 right there. Pardon if I mix up the crossing over letters a little bit, but never mind. Let's work it back through now. The first 3 is ATG, which is going to be AUG. The next 3 it is C, it is GCC. So I'm going to rewrite that. GCC, there's nothing to change. I am only changing the T's into U's. The next 3, it is AAT. So I'm going to have AAU. The next 3, it is GGC. I'm going to rewrite that GGC there. GGC. Then the last 3, it is GAT. So I'm going to change the T into U. So I'm going to have GAU. That's the only thing I've done. Now, the next step is to convert now, which I'm going to say this is translation process. I want my tRNA. My messenger RNA, this is the transcription process. Now, the translation process is when I'm going to convert now my mRNA into tRNA. Technically, it's not a conversion. It's just a reading of it. Okay, so I'm going to call this now tRNA. tRNA. So in the tRNA, what I'm going to do now is to look at the corresponding strand for the mRNA. So the tRNA is the corresponding strand for the mRNA. So please remember that now, okay? So the tRNA is the corresponding strand for the mRNA. So what corresponds to A in the RNA is U. Remember, in RNA, A bond to U, G bond to C, okay? So again, we're going to have here, let's go back one step. So we're going to have U first, U bond to A. And then what bond to U, it is A. And then what bond to G, it is C, God to Christ. Okay, wherever you see G, you see C. Okay, and then you go to the other three right here. So G again go to C, C go to G, so you have two G's. And they go to the other one, which is A bond to U, and then U bond to A, okay? And they go to the other three, G bond to C, and then I think I missed one step, so two C's, and then C bond to G. And then the last one here is going to be G bond to C, and then A bond to U, and then U bond to A. All right, so that's the corresponding, that is the tRNA. So if we look in the table now, we can look for UAC. UAC, it is TRY. Okay, so hey, two of them will be out already. So A will be out because it is not starting with TYR, which is tyrosine. All right, and also C is out. The next one is CGG, and CGG, which is where it is Argine, which is ARG, Argine, ARG. So, okay, so B and C are still in. The third one, which is UUA, so UUA, it is leucine, and so we should have our third be leucine. So D will be out because of glycine right here. It should not be glycine, but what? Leucine. And then if we go to the another one, which is CCG, CCG is proline, beautiful. And then now we have CUA, say CUA is right here, leucine, excellente. B is our correct answer. Excellent. All right, so great. So please, I hope you understand that. That one is a little lengthy for me to stay on and to explain. All right, let's go to question number 19 now. For question number 19, it said, reference to the following diagram not drawn to scale, which shows nuclear material in a cell. All right, for this one, I could go a little bit faster because it said, which of the following shows, following rows matches each structure with its correct name. And so this one, I don't need to explain that because I know you'll know this. For the cell right here, number 1 will represent what they call the chromatids. All right, the chromatids. And then you'll see the double helix right here. So this is DNA. So we know this is DNA right here. All right, so there's no even need to explain that. That's our DNA. And then what holds the DNA structure together is what they call the histone proteins. So these are histone proteins. So 3 is histone protein. And of course, without any doubt, we know that this must be chromosome. Okay, so let's go to the answer now. So A is chromatids, number 1 is chromatins, and right off the bat, that's C. 2 is DNA, 3 is histone proteins, and 4, it is the chromosome. So option C is our correct answer. Let's just move on. All right, excellent. Question number 20. In question number 20, the item refers to the following process of the translation. And this is what we talked about earlier. The translation occurring at the ribose in a cell that synthesizes enzymes. So enzymes are being making right here. Enzymes are proteins, right? So proteins are forming. Now it's said now the structure P most likely represents what? So these round circles, what are they? This, a matter of fact, here this is a tRNA. Let me just put that here. This is a tRNA. The tRNA responsible to transport the amino acids. tRNA responsible to transport the what? Amino acid. Once it reads the code from the mRNA, which is the messenger, it translates it into the protein structure. So what makes the protein structure many amino acids? So this here is the amino acid being made. So the protein structure being made by linking the amino acids together by the peptide bond. So this is amino acids. The circle represents amino acids. So yep. Question 20 is D. All right. Now we can move on now from question 20 to question 21. It said, which of the following statements is not true? Not, the word is not true for most homologous chromosome, which is not true. All right, the first one, it said they are the same length. Typically, they are the same length. So that is a truth. So that's a true statement. Because a matter of fact, homologous chromosomes are grouped together based on size and length, right? Similar size and length. Maybe not be the exact, but yeah, same length. They contain the same alleles. If they contain the same alleles, notice what is happening here, right? That's no variation. But let me talk about homologous chromosome before I even go further for you to understand what I'm going to say now. Homologous chromosomes mean that these chromosomes were taken from both parents. One from the father, one from the mother. Or one from the male, one from the female. So therefore, if they have the same exact alleles, it therefore means that the child will be identical of both parents. Right? In fact, both parents then are typically identical. So the truth and in fact, right, is that containing same alleles cannot be a true statement for homologous chromosomes. The alleles can be different. You can have dominant, you can have recessive. And remember that alleles will be variation or variant form of genes. So definitely B will be your answer. Before even going any further, all right, they contain the same number of genes. Yes, true statement. They contain the same heterochromatin bonds. Yes. So we talk about locus and where the genes are found. Yes. So definitely alleles cannot be the same. If alleles are the same, we'll all be exactly alike. All right. There'll be no changes between any individual at all. All right. Question number 22. All right. So question number 22 now, it said item 22 refers to the following diagram, which represents the relative amount of DNA per nucleus during many cell divisions in an animal cell. And so before you can go anywhere, let's talk about this a little bit. You have N, 2N, 3N, and 4N. Now, if we're talking about reproduction and cell division, right? N represents what they call the gametes, right? Which is haploid. So once you see N, you're talking about haploid, right? Once you see 2N, you're talking about diploid or fertilization. Point that out, okay? So N is haploid, all right? Which means gametes. Let me put or slash gametes, okay? When you see 2N, it represents fertilization or diploid. So that means the cell that formed after fertilization. So this is diploid or fertilization. Let me just put okay, let me write out the entire word. Fertilization. And then now 3N is not something that you'll see in human beings, right? Or typical of organisms. But what you may see is 4N. And what is 4N now? 4N is happening in the interphase of cells when you have the DNA or the chromosomes replicate. All right. So this is replication here. So this is the interphase where you have replication taking place. All right. So replication taking place here. All right. So great. So let's see what is happening here now. For this, so which of the following processes are occurring at X and Y? So what is happening at X and Y moves from 2N into 4N. That means this is after fertilization. This is mitosis taking place right here now, right? To make new cells. All right. And then moves from 2N. No, this is decreasing from 4N into 2N. Okay. So let's see what is happening here now. So X, all right. So when we talk about X again, is going to 4N. So that's 4N is when you have replication taking place. All right, that's the first thing you have to think about right there. So that is when DNA is synthesizing. All right. So X is fertilization. Y is when you have the DNA synthesizing. All right. So this is going to be your answer right here. C is the correct answer. Okay. So again, let me point it out on the graph. So X is when it moves to 4N. That is when you have replication of the chromosomes or DNA. That is the interphase. And Y now is when it is going down to 2N, which is fertilization. So this is fertilization right here. All right, so great. All right, because remember, after cell division, you can mitosis, for example. So when you have meiosis taking place, and then you have a haploid cell, the haploid cell will then be fertilized, and then now you will have your diploid cell again. All right, so great. So that is option C. All right, let's go to question number 23. And this said, item 23 refers to the following information about color in cats. Oh, wow, this is a long question. All right, let's read through it. It's a particular breed of domestic cats can have either black, white, or brown fur. That's 3 colors. Whoa. It said the dominant allele B of one gene gives black fur. So black fur is dominant. And the recessive allele B gives brown fur. So brown is recessive, black is dominant. It said many of the white cats carry a dominant allele P. So it's something interesting happening here now. All right. It's a second gene which inhibits. So P is inhibiting all other pigments. So this is like albino type of stuff, right? All right. It said these recessive allele P has no effect on the color of fur. Okay. It said the 2 different genes are not linked, nor are they on the X chromosome. So no, this is not any sex-linked chromosome on the X chromosome. All right, it said now, it said this type of gene interaction is described as what? And so again, it is not sex-linked because again, it's not on the X chromosome, neither it is specified it's on a Y chromosome. So this is not a sex-linked interaction. Epistasis, let me explain what epistasis is. And epistasis is that when the expression of one gene that suppress the expression of another gene. So please make a note of this. Epistasis is the expression of one gene that suppresses the expression of another gene or trait. All right, so you can make a note of that. And so here look like even the answer because it's saying that P inhibits all pigment production. That mean black or brown cannot be expressed because P is suppressing both of them. So this is like an albino situation. And so this seemed like a good answer right here. I circled B, but B is not the correct answer. I was saying B, it is not a sex link. I don't know why I circled that. So A looks good. B is not. Codominance is not, because with codominance, what codominance does is that they have expression of both traits. So it's like a mixing type of stuff. Say example, you have brown and white, or black and white, or black and brown in that case, right? Dominant and recessive, absolutely not. So epistasis is the correct answer right here, is that one gene suppresses the expression of another gene. All right, 24. In 24, it said it refers to the following diagram showing one stage of mitotic cell division. And here we can say these are our chromosomes. Why I say chromosome and not chromatids? Because a centromere is right here, so it's still a chromosome. All right. It's said no. It's from the position and shape of the chromosomes. Matter of fact, even say chromosomes. What stage of mitosis has been reached? And again, this is what we call the anaphase. Why anaphase? Because the chromosomes are moving to opposite sides of the cells, or opposite poles of the cells. Now, what I want to point out, the easy way to remember is P mat, yeah? So P mat. And if you think about P as in pressed, that means the chromosomes will come together. The M for middle, the chromosomes align in the middle of the cell, which is called the metaphase plate. In the anaphase, antisocial. Don't use the word antisocial in examination. Please do not use it. But I'm helping you to remember it. So when it becomes antisocial, they move to opposite sides of the opposite sides of a cell. That is the anaphase. The telophase, like teleporting, is like they move into their own little capsule. So therefore means that the nuclear membrane are reforming. And that's why you have the cleavage furrow actually forming. But do not use the word antisocial or teleporting in exam. It's just for you to remember what is happening to the chromosomes. All right, so here now it is the anaphase. All right, so let's see what option that is. Yep, and so the option here is C, option C, anaphase. Now, question number 25. All right, and I really need to speed this up because, time. All right, so mutation may arise due to non-disjunction of chromosomes. In which of the following processes would such a mutation be transmitted to the offspring? And when we talk about, firstly right here, because we're talking about non-disjunction. What do you mean by non-disjunction of chromosomes? Now, non-disjunction refers to the failure of the chromosome to separate. So please make a note of that. It is the failure of the chromosomes to what? Separate. Okay. So it's a during division of a cell in the cambium. Not true, because that's not transmitting to the offspring itself. That's just an individual organism will be affected by it. It's a during the division of malpignant cell in the skin. And once ever cell division in the malpignant cell in the skin, that will actually lead to cancer of the skin. All right, not necessarily transmitted to the offspring. After the fusion of male nucleus with the polar nucleus in the plant, this is not too bad, to be honest with you. But let me talk about this a little bit. This is not a typical way for it to transmit to the offspring. In this case, where it occurs after fertilization, what have a condition called the mosaicism. So if it is mosaic, individual will form in this sense. However, now, indeed, during the development of the secondary oocyte in animals, this is the typical way in which the mutation will be transferred to the offspring. All right, is when the secondary oocyte is being formed. And this is when you're forming the egg or the ovum that will be ovulated. So once the chromosomes are not separated here, you'll lead to some conditions such as Down syndrome, right? So this Down syndrome will be the common way to transmit this mutation to the offspring. And not after the fertilization process, because when fertilization will take place, it therefore means that you're assuming that you have the correct number of chromosome in each of the gametes. But during the formation of the gamete itself, once the numbers are not correct and that gamete is being fertilized, then the child will have that mutation. All right, question number 6. Is a possible benefit of producing transgenic plants is what? So when talk about transgenic plant, what are you talking about? You're talking about plants that are being modified. So you can think about, for example, GMOs, for examples, right? These are plants that are being modified. So think about the word trans, change in something. So change in the genes or the genetic makeup, right? So, let's think about the options now. Option A is a determination of human genome, which is not true. Improve human nutrition, that is a good answer there. Let's see if anything beats this, because you can modify the plants genetically to produce more and even to add certain type of nutrients or producing certain type of nutrients within that plant and so on. All right, or even to produce less starch or to produce less sugar in the plant. So you could change the genes to do those things, or even to grow more. It's an increased use of pesticide. Not true, because GMOs could actually reduce the pesticide usage, because you could have GMOs that now are resistant to pest. Production of clones, definitely not true, because if you're changing the genetic makeup, you cannot produce the clones there. Isn't it true? Right? So definitely to improve nutrition will be one of the best answer. Well, the best answer right here. Now, question number 27. Where this is. Right. So question number 27, it said, which of the following events that occurred during meiosis is most likely to result in chromosomal mutation? All right. So we're talking about meiosis, right? And so here now it said non-disjunction. All right. And so it will not be A. All right. And so DNA replication, no. Homologous pairing is a great answer right here, because this is where now let's think about, in this case, let's think about crossing over, for example. Yes. So when you have crossover and synapses taking place, when the chromosomes are pairing up and they're exchanging the genetic materials and so on, if you think about it in that sense, and the mutation taking place of the chromosomes, absolutely. All right, 28. In populations of Pacific salmon, most fish are either very large or very small. This is because sperm from medium-sized fish seldom fertilize the eggs. The allele frequency of medium fish is therefore very low. This is most likely is an example of what? So medium size is very low. No, that's not a typical situation. Let me show you a graph. On a normal population, for any normal population, this or the graph will look. The average size or the medium size will be the most, that is typical of any given population. So let's say you say the average size of person is say 5 feet 7 inches, for example. Then most person will be fall in that category. Persons are shorter than that will be the lower category. Person may be taller, maybe in higher category, and so on. So now, what is important now? The medium size. So therefore now, what this statement is saying is that the small fish is high, and then the medium size are low, and then the large fish are high. So this will represent the medium, this will represent the small, and this will represent the large. So when the medium size are smaller, what is taking place here is called what they call a disruptive selection. All right, not artificial selection, because artificial selection you select the one that you prefer. So you may select the largest one to and so on. All right, stabilizing, of course, you have a normal population. Directional is when you have a parallel in terms of the changes. And so disruptive selection is definitely the answer, because disruptive is when the changes are favored on the extreme ends of the traits. So example, the extreme ends here, small and large, it's not favored for the average or the medium size. All right, so the medium size is not favored by disruptive selection. All right, so now let's go to question number 29. I have a question number 29 refers to the following map of a plasmid showing the position of recognition sequences of 2 restriction enzymes. So you have 2 restriction enzymes right here. So you have the genes for the ampicillin resistance, and then you have here the gene now for the tetracycline resistance. And so you have the BAM-H1 here and the EcoR1 right here. And so very important, let's read through this, matter of fact. It said, in addition to the human insulin gene, which of the following enzymes would be required to produce a recombinant plasmid? Recombinant plasmid. Without going any further, no. This is what you need to think about. Recombinant refers to the combining of. You're recombining. Yeah? So if you're going to recombine, what enzyme do you need to recombine this plasmid? And this is the DNA. So you're talking about the DNA ligase. The ligase is the enzyme that will be representing the combination or recombination of the strands itself. So the DNA ligase are responsible for joining of the strands. All right, that's very important, joining of the strands. All right, question number 30. I'm kind of go a little bit fast in order so quick I could finish this. So question number 30, right, is said here, which type of isolation plays the most significant role in allopatric speciation? Now, when you talk about allopatric speciation, you're talking about a barrier that's separating them based on their location. So there is a geographic separation or barrier. Now, some examples of geographical separation or barriers, we're talking about like rivers, like mountains, even desert, lake, anything that separates a body of land technically, or create different environment in between the 2 different geographic locations, then yes. So example, talking about land animals, they cannot cross water, for example. So the big body of water like the ocean or the river or the lake will prevent them from crossing over. So the isolation right here will cause by a geographic, is geographic. Let me just delete this real quick. Right, that's just a simple mistake there. Sorry about that. So this is our answer here, A. Now 31 is that which of the following structures are formed during the development of pollen grains? And let me just quickly run through this. I I want to spend some time to explain this, but let's see how quick I could do it anyways. All right, when we talk about antipodals, right? The antipodals you're talking about now, they provide nourishment for the cell. All right, typically. When we talk about the synergids, they also secrete chemicals that help to guide the pollen tube. Just to make a note of that. So the synergids, they secrete chemical, they produce and secrete, a matter of fact, chemicals that guide the pollen tubes. All right, and then now when we talk about the polar nuclei, the polar nuclei will fuse with the sperm cell from the pollen grain to form the endosperm itself. All right, and so here, the pollen tube will be answer, matter of fact, right? So the pollen, the tube nuclei will be your answer, because the tube nuclei will head in form what they call the pollen tube. All right, and so that is important to form during the pollen grain development. All right, so when the pollen grain is developing, the tube nuclei will be developed that will form into what they call the pollen tube. All right, the other structures will be there already. Now, item 33 refers to the following diagram, which shows the embryo sac of a flowering plant. And so I might as well label these. And these structures are the same structures that I talk about up top there. So I must point out where they are. So this will be the antipodal right here. Antipodal. Right, that's the antipodal right there. And structure 2 will be our polar nuclei. All right, or our polar bodies. The polar nuclei right here. Okay. Polar nuclei. All right, so, or the polar bodies. All right, so here now 3 will be the synergids. All right, so synergids will be here. Synergids. All right, will be there. There's a synergid. All right, and then 4 will be a vacuole. All right, great. All right, so synergids are down here and the antipodals are on the top right here. All right, so there are 3 antipodals. All right, so, okay, and this here now. All right, and right here, that is not labeled, there's no line here. This is a central cell, a matter of fact. So this is where you find the central cell. All right, so great. And you can check out, I have a video or lesson on pollen grains. You can check that one out. That will kind of help you with the pollen grain itself. Just some basic stuff generally. All right, question number 32. All right, it says which of the label structure develops into the endosperm? And the endosperm will be, as I mentioned before, will be the polar nuclei, because the polar nuclei again will be fused or fertilized by the sperm cells and form the endosperm. So definitely endosperm is a polar nuclei, which is structure 2. Structure 2 is our polar nuclei, right? Which is the structure right here. Yes. All right. So question number 33. And question number 33 reads, which of the following terms is most appropriate to describe a plant in which the anther of the flower mature before the stigma? So the anther mature before the stigma. This now is in the male part matures before the female part. And so let's now point out these words really quick. And so here now, this is actually protandry is the answer here, because this is the correct one here. So protandry is when the male part develops before the female part. So protandry is the correct answer. In terms of protogeny, which is the opposite. Protogeny is opposite of protandry, and protogeny is when the female part matures before the male part. Okay, so I can make a note of this. The opposite of protandry, male part. So protandry is male before female. All right, so now we'll say here protandry, male before female. I'm going to talk about maturity. So male before female, just to make it short. Okay, they can always write that out when you're reviewing this. And then the protogeny is when the female before the male, matures before the male. Okay, and we're talking about the reproductive part of the flower. Okay. All right. So great. And then now, for part C and D, they're the same word. All right? Monoecious and hermaphrodite, they mean the same thing, which means both male and female part. And as a matter of fact, if you have both of them there, they could not be the answer anyhow, right? All right, so both male and female parts, a flow containing both male and female parts, and of course, development would be similar. All right, number 34. Which of the following statement is true about double fertilization in flowering plants? I went about double fertilization. You're talking about, matter of fact, before we even go any further, let me just make this statement right here. This is something that you need to know. One pollen equals to 2 sperm cells. Yep. One pollen equals to 2 sperm cells. That's the first thing to know. So a single pollen grain releases 2 sperm cells. So in this case now, one of the sperm cell will fertilize the nucleus of the egg or the ovum, and then now one will go to fertilize the polar nuclei. And the polar nuclei, when it's fertilized, will form the endosperm. Please make a note of that. So double fertilization is when both the egg or the ovum is being fertilized along with the polar nuclei to form the endosperm. All right, and this is in plants, a matter of fact, just to make a note of that, right? So option D look like our perfect answer right here. So 2 sperm nuclei, definitely coming from one pollen. It fused with an egg, so an egg is being fertilized along with a polar body. So they do this at the same time. All right, that's what we call double fertilization. So the egg is being fertilized, and of course you know that when the ovum fertilized, they form the seeds, all right? And then the polar nuclei, when they divide, they form what they call the endosperms. All right, so an endosperm is different in monocot and dicotyledon, and we could talk about that another time. All right, but let's go to question number 35. I'm trying to go as fast as possible now. In 35, it's the item 35 refers to the following diagram showing a mature pollen grain. And this is a pollen grain. I think I need to label this for you real quick. This bigger one right here, this is what they call the generative nucleus or cell. So this is generative, generative nuclei, all right, or cell. And this one over here is called the tube nuclei. Okay, so this is a tube nuclei. All right, and let's put nuclei beside this one as well, just to make sure we have them in the right place. All right, and part 3 right is called the enzyme. So this is enzyme. Again, you can look on my my video on pollen grain. I show you these diagrams and the labeling for them. And this is the intine, just to make a note of that. All right. All right, so here now it said which of the labeled structures will give rise to 2 male gametes? 2 male gametes. And so if we're going to have 2 male gametes right here, right? All right, our answer must be our generative nuclei. Okay, so the male gametes, which is the pollen grain. So just to make a note that generative nuclei forms the pollen grain. That's where the entire pollen grain is right here. But in the pollen grain, the generative nuclei will form the sperm cells, right? Just to make a note of that, form the sperm cells. So option D will be your answer right here. The tube nuclei now will form the pollen tube from the pollen grain. So answer here has to be option D, yeah? All right, great. So again, generative nuclei forms the sperm cells. The tube nuclei will form the pollen tube, and that's when you have after pollination, the pollen tube will form from the tube nuclei. All right, and these go space right is called the pollen pores. Okay, just to make a note of that. All right, let's just quickly, so fast I could go to question number 36. In question number 36, which of the following condition does not promote cross-pollination? Does not promote cross-pollination. And so even by going any further, cleistogamy, right? So cleistogamy is what they call self-pollination. So if it's self-pollination, it can't promote cross-pollination, right? So it does not promote cross-pollination because it itself, it is self-pollination. All right. And so this is self-pollination. Just to make a note here, this promotes self-pollination. Self-pollination. All right, so just make a note of that real quick. Again, a protogeny, again, refers to when the female part matures before the male part, and protandry is when the male part matures before the female part. We just talked about that earlier, right? And so it doesn't matter, any of them could aid in cross-pollination, right? All right, great. All right, and of course, self-incompatibility, not compatible for self, mean that it must be something else. So therefore, yes. All right, great. Anyway, question number 37. Let's jump into that and just move on real quick. In plants, the ovary is best described as the what? Ovary. Let's think about the ovary. The tip of the carpel, what is that? Is not the stigma. Yep. So the tip of the carpel is what they call the stigma. So it is not the ovary. B, the basic unit of the gynoecium, which is not, because we're talking about just a single unit. And here now we talk about the solid base of a single carpel. And okay, you know what? Let me just show you. So it's a solid base of a single carpel. Let me draw this. I guess you see it better if I do this because C look good. So I'm going to draw here the carpel, and the carpel has 3 parts. So I just want to draw all of that. The carpel has, I ignore that line. I'm going to erase it anyway. So there are 3 parts in a carpel. You know what? Let me erase this and make it a little better. All right, need a little better than that. But there are 3 parts of a carpel: the stigma, style, and ovary. The carpel is also known as the pistil, okay, which is the female part of the flower. Okay, so again, you'll have the stigma. This is a better diagram now. And then coming down with the style, then the ovary at the base right there, going back up. Right, perfect. So here now, as I mentioned, we have here the stigma, which is right here. So stigma right here, the style, and the ovary. So the carpel now, again, is the female part, which is the whole entire structure right here. So the swollen base, this base, the swollen base is the ovary. So the swollen base of the carpel is the ovary. The female reproductive part of the flower is the same as the carpel, which is the entire thing. So the ovary can't be the entire thing itself. It must be only the swollen part. And in this part you have what they call ovules. So you have some ovules in here. And what you need to remember that the ovary forms the fruit, right? And the ovule forms the seed. So next time eating a fruit, you know what you're eating? A developed ovary, okay? So you can say you're going to the food store to buy some developed ovaries, which are fruits indeed. All right, question number 38. In question number 38, it says, which of the following statements identifies a difference between the secondary oocytes and the ovum? So when you talk about the secondary oocyte and the ovum, first you need to think about is that the secondary oocyte, you're talking about just before ovulation, right? Just before it ovulates. So here now, firstly, right, is that what I need to think about is that the primary oocyte, just to make a note of that, you talk about it's diploid. I just want to make a note of that, all right? That the primary oocyte is diploid. The secondary will be haploid. That's when it divides, get an ovum, is just half number of chromosomes, but the primary is diploid. I just want to make a note of that. All right. Also, what is important right here is, let me just run through quickly. So the secondary oocyte is diploid, while the ovum is haploid. Not true. Both of them are haploid. So I want to take this thing out. The only one that is diploid is the primary oocyte. Okay, let's take this piece out. All right, great. So both are haploid. Next here is that the secondary oocyte has 2 polar bodies, while the ovum has 1. Not true at all. They are both technically the same. It said the secondary oocyte is surrounded by a zone of peculia, while the ovum is not. Both of them are. D is the secondary oocyte is at the metaphase 2 of meiosis, while ovum has a completed the mitotic cell division. Absolutely, because this is what will be ovulated, right? That is what will be ready for fertilization. So definitely it is completed, and D is our answer. All right, question number 39. We're almost there, gentle people. 39. It says 39 refers to the following diagram of the human female reproductive system. Let's see how quickly we could go through this. All right, so part 1 here, this is our ovary. So here is our ovary. Then we have a fallopian tube or oviduct. So ovary is right here. I just like to be detailed a little bit. Fallopian tube. Fallopian tube is also the same thing as fallopian tube is the same thing as the oviduct, just to make a mention of that. The fallopian tube is the same thing as oviduct. Part 3 is the uterus. I want to say our uterus. I don't have uterus. All right, females have uterus, but it's the human uterus. All right, and part 4 here is the vagina. All right, great. So it said now, at which of the regions, label 1, 2, 3, and 4, does fertilization takes place? And this one's a direct one. It takes place in the fallopian tube, so therefore this has to be B. All right, question number 40. In question number 40, it said human spermatozoa have to spend several hours in the female genital tract before they can fertilize an oocyte. Oh yes. During this time, the spermatozoa are activated in a process called what? And so while they're in there, remember now that going along the female passage, because of the harshness of the passage, some of the sperms will eventually die. All right. And so what will happen here now, that the sperms will actually change, right? Will actually change so they can now penetrate the egg itself to cause fertilization. Now, that change in the cell or the sperm itself is called a capacitation. All right. So this here now is a process that is taking place right here. Okay. And so ejaculation is a release of a sperm from the male penis. And so therefore it cannot be, as it's noticed here, spend several hours ejaculating inside the female genital. That's not even making sense. So ejaculation is not true. Conception is where after fertilization takes place. That means, well, technically it's the process of fertilization. You conceive now. That means the egg is being fertilized and implanted. And copulation is the act of the male and female going together to reproduce and all of that, right? So yes, by even process of elimination, definitely A is the correct answer. All right, 5 more questions to go. Can you believe it? Let's run through these 5 questions real quick. So question 41. Here now, it states that which of the following events during the ovarian cycle is a result of the negative feedback caused by an increase in estrogen level. Now, the first I want to point out, you see this right here? Negative feedback. What negative feedback means? This is the first thing to think about before you even jump into the question and into the answers. Negative feedback means now as one increasing, the other one is decreasing. So looking for as something increases, the next one decrease. So you need to think about now what is increasing. Matter of fact, the question says estrogen is increasing. So what do you need to think about what is decreasing while estrogen is increasing? So what you need to remember is the menstrual cycle graph and think about it. So it's a decrease in production of progesterone at the end of the secretory phase, which is not true, because if progesterone decrease after the secretory phase, right, it therefore means that menstrual cycle will start, and of course estrogen will kind of be a little bit low at that point. Yeah, so it cannot be true. For B now, it's an increased secretion of the luteinizing hormone during the follicular phase. And the luteinizing hormone, again, if you should remember, it is responsible for the formation and development of the yellow body. All right. And it's also very important as well. That falls after ovulation. All right. That is when estrogen is already high. All right. And so for part C now, it's an increased secretion of the gonadotropin releasing hormone, which is the D, which is a GTH hormone here, is during the follicular phase, which is not true. This is not true on this phase at all. All right, so D now is a decrease in the secretion of follicular stimulating hormone, which is FSH, after the release of a secondary oocyte. Yes, this is indeed correct answer right here. All right, so importantly, yep. So definitely the HCG hormone, yep, which definitely will be right here. All right, awesome. All right, so 42. I'm jumping a little bit too fast now. 42. All right, so 42. A sample of woman's urine is tested at the laboratory, and human chronic gonadotropin is detected. Okay, so this is now HCG hormones right here. Just to make a note of it, so if you see the HCG hormone, you're talking about the human chronic gonadotropin hormone. All right, HCG. All right, so great. So what this indicates, and this is a measure. Remember that this is when a female is taking a pregnancy test, yeah, and she will urinate on that test, and if it show that she's pregnant or not, so it just show that pregnancy is taking place. It's a test of pregnancy. And so what indicates pregnancy is not ovulation as occurring. You can ovulate and not be pregnant. So this is not true. So A cannot be correct because you can ovulate and not become pregnant. It said the menstruation began. If you're menstruating, you're not pregnant. So this is not correct. If you're menstruating, you're not pregnant. Estrogen levels has decreased. If estrogen levels has decreased, is when menstruation gonna take place as well. So definitely implantation of the embryo has occurred. That means once the embryo implanted, that means you're sure that you're pregnant, right? Because yes, a fertilized egg has been implanted, and it's an embryo. Embryo means that zygote was formed, and after the zygote formed, then it started developing into embryo. So now we show that the pregnancy has taken place. All right. So item 43 and 44 refer to the following hormones. You have estrogen, progesterone, luteinizing hormone, follicular stimulating hormone is FSH, and you have LH. All right, so now we said 43, it said which hormone stimulates the development of the endometrium, and this is the estrogen. Estrogen stimulates it, progesterone thickens it. So this is option A right here. It said which hormone regulates the precise timing of ovulation, and this is done by the luteinizing hormone, which is LH. So this is C, definitely. And 45, and we are finished. It said item 45 refer to the following diagram showing a transverse section through the middle piece of a sperm cell. And so let me quickly label this for you, and I'm out of here after this. So this structure right here, let me put the lines first. This is the mitochondria. These here will be fiber, and this here H will be the microtubule, and these here will be our intersheath. So let me just quickly label this. So this is mitochondria. Remember that sperm cells have a lot of mitochondria in the middle piece. Do you remember why? Is to produce a lot of energy, so the sperm could swim. So you can make a note of that. Okay, so in the mid piece, there's a lot of mitochondria to produce a lot of energy, so sperms can swim. Okay. So this is mitochondrion. That's one of them. This is the microtubule. This H is microtubule. Microtubule. All right. All right. And this here is the inner sheath. Inner sheath. All right, great. And then this now will be here. It is actually fiber. All right. So great. And so the question now asks, the structure label H is, and that is microtubules. And general people, thank you for being here. Please do well on the examination. See you next time.